Welcome to our deep dive into food safety and antibiotic resistance. Salmonella, a leading cause of foodborne illness worldwide, is increasingly found in animal-derived foods. Dr. Gibehu, can you explain why Salmonella is so concerning, especially in contexts like Ethiopia?
Certainly. Salmonella isn't just a common foodborne pathogen. It's notable for its zoonotic potential. It can jump from animals to humans through products like milk and beef. In places where raw or undercooked animal products are culturally favored, such as Ethiopia, the risk of widespread infection and resistance grows exponentially.
That's a critical point. The WHO estimates millions of Salmonella cases each year globally, often leading to severe gastroenteritis or worse. How does the cultural practice of consuming raw meat and milk drive these risks?
In Ethiopia, dishes like kurt and kitfo involve raw or lightly cooked beef, and raw milk is widely consumed. These traditions, combined with insufficient hygiene in food preparation, create an ideal environment for Salmonella transmission and, alarmingly, for the spread of multi-drug resistant strains.
Let's move to the recent study in Sidama region, which focused on Salmonella in cow, milk, and beef. Could you outline how researchers structured their sampling and why these areas were chosen?
Absolutely. The districts Hawassa, Yirgalum, and Wando Genet were selected due to their high milk and meat production. Researchers used a cross-sectional design, collecting 216 samples from retailers, farms, and households, ensuring a representative snapshot of the region's dairy and beef supply chain.
Interesting. By encompassing sources from commercial farms to small households, the study could assess not only prevalence, but also differences in hygiene and handling practices, correct?
Exactly. The mixed sampling approach provided insights into how various handling and production practices might affect Salmonella contamination, allowing comparisons across the food chain.
Turning to laboratory methods, how did the researchers detect and confirm Salmonella in these samples? I understand both traditional and molecular techniques were used.
That's right. Initially, samples underwent standard bacteriological culture to isolate organisms resembling Salmonella. These were then verified biochemically, but crucially, confirmation relied on PCR targeting a 496 base pair segment of the histidine operona, highly specific genetic marker for Salmonella.
So, why is PCR considered more reliable than conventional methods in this context?
Conventional biochemical tests can misidentify related bacteria. PCR, by amplifying a Salmonella-specific DNA segment, offers higher specificity and sensitivity. However, it's not immune to technical errors like contamination or pipetting mistakes, so rigorous controls are essential.
Let's discuss the findings. What were the key prevalence rates for Salmonella in the different sample types and districts?
Salmonella was isolated from 4.17% of all samples based on molecular testing. Prevalence was similar across the three districts: Hawassa at 5.38%, Yirgalum at 3.23%, and Wando Genet at 2.38%. There was also little difference between milk, 3.9%, and beef, 4.7% samples.
That suggests contamination is a widespread issue, not limited to particular areas or product types. Were there significant differences based on source, such as retailers versus farms?
Not really. The rates didn't vary significantly between retailers, households, and farms, indicating that similar hygiene practices or lack thereof persist throughout the supply chain.
Now, the crux of the matter: antibiotic resistance. What did the susceptibility test reveal about the Salmonella isolates from these samples?
Alarmingly, every Salmonella isolate was resistant to ampicillin, nalidixic acid, clindamycin, and cephalothina. 100% resistance rate for these drugs. Sensitivity to other antibiotics like ceftriaxone, 88.9%, chloramphenicol, 55.6%, and gentamicin, 66.7% varied, but all isolates showed multi-drug resistance, MDR, resisting at least 5 antibiotic classes.
That level of MDR is concerning. How does this compare to previous studies or global trends?
These findings actually surpass some previous Ethiopian studies where MDR rates ranged from 31.8% to 52.5%. It mirrors a global trend of increasing resistance, likely driven by widespread and sometimes inappropriate antibiotic use in livestock and human medicine.
What do you see as the root causes of this MDR phenomenon in Salmonella, particularly in the Ethiopian context?
Several factors converge here. First, antibiotics are often overused as growth promoters in livestock or given without veterinary oversight. Second, limited regulation and access to proper veterinary care mean drugs are administered haphazardly, creating selection pressure for resistant strains. A similar pattern has been observed globally, but it's exacerbated where oversight is lax.
Could you elaborate with an analogy for our listeners?
Imagine a garden overrun with weeds if you use the same herbicide repeatedly, only the resistant weeds survive. The same happens with bacteria. Repeated exposure to the same antibiotics allows only resistant strains like these Salmonella to persist and multiply.
Given these findings, what are the major public health implications, and what steps should be prioritized to mitigate the threat?
The presence of MDR Salmonella in food chains means infections may no longer respond to standard treatments, leading to longer illnesses, higher healthcare costs, and greater risk of outbreaks. Key interventions include strict hygiene at every step of the food chain, prudent antibiotic use, and robust training for farmers and handlers.
Would public education or regulatory changes make a difference?
Absolutely. Public education campaigns, especially about the risks of consuming raw animal products and the importance of proper food handling, are crucial. Regulatory bodies must enforce guidelines on antibiotic usage and food hygiene, ideally in tandem with regular microbiological surveillance.
Before we close, what research gaps remain and what lessons can be drawn for other countries facing similar challenges?
Further studies are needed to determine the specific serotypes of Salmonella and to identify the genes behind resistance. Detailed epidemiological tracking could reveal how MDR strains spread. Globally, this Ethiopian case underscores the universal need for integrated surveillance, responsible antibiotic stewardship, and cross-sector collaboration, what public health experts call a One Health approach.
A comprehensive message. Thank you, Dr. Gebiehu, for unpacking these complex issues and sharing practical insights on tackling salmonella and antimicrobial resistance in food systems.